Okay, folks, so rather than kind of keep you too long, and you know that the course is a little bit different because the way that you're going to be evaluated is all going to be where you have your notes in front of you. It'll kind of be like an open book, but it's also like all online. Not sure how you're going to do it because you don't have a computer. Do you have access to one?
Yeah, I have one at home.
Okay, I can lend you one if you want. So it's a little bit different. So I want you to kind of sit back, hear a little bit about the experiences that James and Zed have had, and then ask them questions because you're not going to be tested in the traditional way. As long as you've kind of been paying attention to the discussions that we're having, I'm hoping that it gives you different ideas about your career path and journey if you're not sure. And then it also gives you an opportunity to ask about individual experiences. Because we are having some technical difficulties and the technician is coming from another part of campus, what I think we're going to do is we're going to try to get through James's talk first. He's going to talk you through the slides. So if you have a copy of the slides in front of you, he'll tell you like it's slide number 1 or the graph on slide number 3, and then we'll try to describe it. I am going to record it, and I have their permission to record it and to keep it locally within the class. So if you've missed something, feel free to ask and then go back to the recording later. Is that okay for everyone? All right, so let's help welcome James to our class.
Hey guys, so thanks for having me here today. So I guess we're kind of going to improvise here with just a talk. I do apologize, I did change up the slides since I sent it to Dr. G yesterday, but I will send them the updated one and I'll annotate it so you guys don't miss anything either at the same time. So a little bit about myself. So I'm a 4th year grad student at Dr. Cynthia Hawkins' lab at SickKids. So I graduated in 2016, and the fall of 2016 is when I started my grad school. Last year, or early this year, end of last year, I joined a startup I formed a startup with one of my lab mates called BioBox, and I'll kind of also go through that later on with you guys on why we started it and what we kind of do as well. And a little bit about my future and kind of the path it's taking, and it's a little bit different than what you guys will probably expect. So kind of to start on the research that I've been doing for the past 3 years. So in terms of first looking at cancer as a whole, so approximately 200,000 new cases of cancer is diagnosed each year in Canada, and about 1 in 2 Canadians will develop cancer in their lifetime. So the challenge that researchers and clinicians are facing today is improving the diagnosis, finding preventative measures in terms of such as lifestyles or certain kind of medications that they can give, as well as management and treatments of the disease. So our lab in particular focuses on pediatric brain tumors. And so when we look at the pediatric population, we see that the tumors of the central nervous system make up a significant proportion of the tumors that we see in those group of patients. That's in the little bar plots that you see in slide 4, I believe. Titled tumors of the CNS and high frequency in pediatric populations. And though it's not the most common type of tumors that we see in these populations, we see that the tumors of the central nervous systems are the leading cause of death in these pediatric as well as adolescent population. And part of the reason for this is because leukemia, which is the most common types of cancers that we see in pediatric patients, research has progressed quite a bit. As you can see in the next slide with the 2 pie charts and the linear graphs at the bottom. So rate of survival of leukemia has increased over the years due to the progression in research. However, brain tumor, in terms of progress in brain tumor research, we haven't gone very far. And so part of this reason is a lack of a targeted therapy to treat these tumors. As well as the location of the tumors. So because these tumors occur in the central nervous system, so they're, for example, one of the tumors that we kind of focus on is diffuse intrinsic pontine glioma, which are tumors that occur in the brainstem. And so to go in there and surgically resect those tumors, it's very difficult because of the sensitive location of those tumors. Not only that, for the patients that do survive and lead to remission of these tumors, there is impact of these non-specific therapies which include radiation and chemotherapy. So a lot of these patients that grow up and have or try to have a healthy lifestyle, they do feel the effects of those treatments, such as psychosocial symptoms, impaired growth and development, cognitive dysfunction, endocrine dysfunction, and more. And so there's really a need to develop specific and personalized treatments towards these tumors that occur. And so to kind of lead that way, sequencing efforts have been underway to molecularly characterize these tumors to understand how these tumors are being formed and what is really driving the formation. And the work, some of the work that our lab and other labs have found over the past few years is that mutations in the histone genes form a significant population of these brain tumors. So a little bit of a review from HMB422. So histones are the protein components of the nucleosomes that organize DNA into chromatins. So the H2A, H2B, H3, and H4 form octamers in which the DNA wraps around and organizes the genome. And what's unique about the histone is that it can play various different roles in the genome, and has a lot of epigenetic roles. So and part of the reason is because of its N-terminal lysine residue, which is subject to post-translational modification. So depending on whether these histones are acetylated or methylated on different regions of its N-terminal motif, it can have different effects on gene regulation, DNA repair, chromosome organization, and the list kind of goes on and on. So trying to understand how these mutations in these histones are driving tumors is one of the challenges that our lab is facing. In terms of histone mutations across cancer, we see that it occurs in more regions than the brain. Obviously, occurs at a very high percentage in tumors of the bones, and we see them in head and neck tumors as well too. The one that I've been focusing on mostly is the H3.3 G34R mutation, which occurs in the hemispheric regions of the brain. And so not much has been characterized, and so that's the role that I kind of took on as a grad student when I started in Cynthia's lab. So going on to the slide saying surveying the interaction with the histone H3.3. So we decided to take a bottoms-up approach to look at these tumors, and so bottoms-up meaning looking at mechanisms and the effects of this mutation and how it affects normal cellular processes. And so we used a method called BioID. And so BioID is where we express our wild-type or mutant histones as a fusion protein with a biotin ligase called BIR-A. And so when we express this fusion proteins in our cells, the biotin ligase that is attached to our histones will biotinylate proteins that are in near proximity of this protein. So when we express these proteins and treat our cells with biotin, we get biotinylation of proteins that are interacting with our wild-type and mutant histones. So when we do a biotin affinity capture and pull down these proteins, we can do mass spec and look at the enrichment of proteins that are interacting with either our wild-type and G34R, and see how this mutation is affecting the interactome of this histones. And so when we pull it down, we see that about 170 proteins are lost or losing interaction with our wild-type and our G34R. So what we did was kind of speeding through it because I guess I had a lot more stuff than I thought. So we did a gene ontology analysis. And so this is where we look at the list of the proteins that we pull down, and we look at different pathways that are making up a significant proportion of this list of genes that we identified as interacting with G34R. And one of the ones we found that was losing interaction was DNA repair proteins and the double-stranded repair pathway. And so it's hard to kind of explain over words, but I'll send you guys the annotations. So when we look at double-stranded break repair, there's kind of two major pathways. So there's the classical non-homologous end joining pathway and homologous recombination. So NHEJ is the error-prone repair pathway, and HR is the classically the error-free. It's not as clear-cut, but that's kind of the point that I wanted to get across. And so we see that that's dependent on the methylation status of our histone H3.3. Okay. 'Cause, 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 Okay, so is there a way I can pull up the laser point?
Let me see if I can.
No. Just Okay, sorry. Depending on the methylation of status of these histones, as I said, that affects the roles that it plays. When it's trimethylated, we see that it leads to error-free homologous recombination, whereas when it's dimethylated, it leads to classical non-homologous end joining. So based on the list of proteins that we pulled down from the bioID, we saw that our mutant histones were losing interaction with proteins involved in the error-free homologous recombination over here. So to kind of look at the repair of these DNA, we did something called a COMET assay, which is where we treat our cells with radiation that causes double-stranded breaks, and we looked at the repair of those genomic DNAs over time by cover slipping those cells or plating those cells at different time points after radiation and running them through gel electrophoresis to look at the migration of DNA. So the more the DNA migrates, the more broken the DNA is. So initially when we treat the cells with radiation, we see that we get a very long tail, which is associated with those broken DNAs. And over time, we see that those DNAs tails get shorter, which is showing the repair of those genomic DNAs. And so when we compare cells that are expressing wild type and G34R, we saw that G34R cells were a little bit slower in repairing those genomic DNA post-radiation. And this occurred at a much later time post-radiation, about approximately 3 hours, which has been associated with a defect in homologous recombination repair because it is a slower process. To kind of look at the mechanism of it, when we look at homologous recombination in the context of histones, we see that, as I mentioned before, that SETD2-mediated trimethylation of these histones leads to recruitment of LGF and then downstream homologous recombination and repair proteins. So I wanted to look at the recruitment of these proteins to see if they were affected at the mutant histones. And so to do this, we used a method called proximity ligation assay. So this is a immunocytochemistry experiment where we label the two proteins of our interest that we're looking to check their interaction using two different antibodies. And those antibodies will subsequently be labeled with secondary antibodies conjugated to an oligonucleotide. And these oligonucleotides, when the two proteins are in close proximity, can ligate together and be amplified and labeled with a fluorochrome to look at the interaction of these proteins. So when we look at the cells, one foci would suggest one interaction between those two proteins, or a complex of those proteins. So essentially, in my experiment, I cover slit the cells and I checked the interaction of these either wild type or mutant histones after radiation or before radiation to look at the response and the recruitment of these repair proteins. Just to kind of make sense of the graph, so y-axis is representing foci per nucleus, so foci would be one interaction as we kind of interpret. And we looked at the wild type versus mutant histone expressing cells with and without radiation, and we see that with radiation, wild type has an increased interaction with RAD51, which is associated with the error-free homologous recombination repair pathway, whereas the mutant histones actually lose interaction with those RAD51, which suggests that they're using the more error-prone non-homologous end joining pathway. These are representative images kind of shown on the side. And we see that we went through the proteins that are important in the early signaling. So SETD2, we also see that G34R has a decreased interaction compared to wild type, as well as that GF, we see essentially no interaction between the G34R compared to the wild type. So this all suggested that there was a defect in this homologous recombination pathway, and so what I wanted to check next was if there was this kind of compensation through the error-prone non-homologous end-joining pathway. And so when we look at one of the key markers, 53BP1, we see that G34R has an increase in higher interaction with that protein post-radiation compared to wild type, suggesting that there may be a compensation happening in these cells through this error-free non-homologous end-joining pathway. And these were all kind of cell line models that I was looking at to also check if this effect was potentially happening in clinical samples. We looked at the sequencing data that we had on clinical tumors just to look at the mutational burden, and we did see that G34R tumors did have a higher mutational load compared to wild type or K27M mutated tumors, as well as carrying mutational signatures associated with the defect in double-stranded break repair, suggesting that this may be applicable in clinical tumors. So why do we care about this? So I hope I convinced you that there is some form of defect in DNA repair in these cells, but this is important in terms of therapeutically targeting these tumors. And a prime example, we see this in BRCA1 mutated ovarian or breast tumors. So because these tumors, when these tumors have a mutation in the BRCA1 genes, which is important in the homologous recombination pathway, they compensate the repair of DNAs through pathways in terms of base excision repair. And these tumors are commonly targeted using PARP inhibitors to sensitize these tumors to genomic instability, and therefore being able to target these tumor cells without affecting the normal cells. And so my research going forward for the next few months will be to look at if I can target these double-stranded break repair pathways to reduce the viability of these G34 expressing tumor cells compared to wild type cells. We're making good time now. So that's the kind of the work that I've been going on. I've been doing at my lab. So on the flip side, as I was doing my research at SickKids, what I didn't expect was that there would be a ton of bioinformatics involved in cancer research, and so it was a bit of a surprise as well as a bit of a challenge to take on research involving those kind of next generation sequencing analysis. And we saw that with a lot of grad students as well. So because of that, it kind of inspired me and one of my lab mates, as well as others, to form this company called BioBox Analytics. And so just to kind of give you a big overview, so our mission as a company, as a startup, is to analyze genomic big data and increase access to these kind of experiments to allow for data integration and analytics, as well as distribution and publication. These aren't in the slide sets that I sent to Dr. Ju, but I'll post these up after.
Sorry, James, could I ask, so what was your background, your bachelor's degree in?
Bachelor's degree was in pathobiology and neuroscience.
So did you have a lot of background in like analytics and doing gene sequencing when you were in undergrad?
Nope, I had to learn all of that in grad school. And I don't know what your guys' undergrad experiences were, but I felt like a lot of the new grad students also in our lab, they had very little experience with this, and so they had to kind of learn this from ground up. And I was kind of lucky to be at SickKids because they do have these next generation sequencing boot camps where they actually teach you how to analyze and look at the data and kind of understand it. And so I felt lucky to be at that institution, but coming out of undergrad, I definitely was very overwhelmed early on in terms of looking at these kind of data. So what is bioinformatics? So bioinformatics is kind of the combination of statistics, computer science, and biology. Sorry, it's not sure. And so it's become a big part of biological research due to the advancements of next generation sequencing. So how many of you guys are familiar with next generation sequencing technologies and techniques? Not very many. Okay, so I have a little bit of background slides so you guys can kind of get a bit of a context as well too. This this graph is kind of showing proportions of papers that are published over the past 20, 30 years with sequencing in it. We see that initially there's a bit of a jump after the advancement of Sanger sequencing, which is the low throughput sequencing. The human genome sequencing was completed in 2000s, and next generation sequencing platforms were developed in 2005. And since that happened, you can see kind of an explosion in the number of papers and research being done using these next generation sequencing technologies. And so if you guys are considering doing research in the future, whether it's in microbiology, cancer biology, neuroscience even, next generation sequencing is going to be an important component of it, I would say. So just to kind of introduce the theory of next generation sequencing. So we have first generation sequencing, which is a Sanger sequencing. And so when we look at a regular PCR reaction, for example, so we have our template DNA in the black over here. So we treat the template with primers as well as DNA polymerase and deoxynucleotide triphosphate, which allows the DNA polymerase to extend this chain of primers using DNTPs, so you get amplification of your DNA strand. So Sanger sequencing kind of takes that, uses that technique with the addition of dideoxy-NTPs. So what happens is when these dideoxynucleotide triphosphates are incorporated, the DNA polymerase is not able to amplify the strand anymore. So depending on the random integration of, sorry, these are meant to be ATGC, I should change that. But depending on the random integration of these fluorophores along with random deoxynucleotide triphosphates, you get random stopping of the amplification of the template of strands. So, and these dideoxynucleotide triphosphates are labeled with fluorophores, and so when you run this on a gel, you can look at the fluorescent signals and kind of infer the sequence of your templates. And I kind of went through it really fast, but there's a lot of really good videos on YouTube that you guys can kind of look up, and that'll explain it really well as well too. So next generation sequencing is essentially the application of Sanger sequencing in a massively parallel fashion. So the technologies has gotten a lot better and better, and so essentially we can get sequencing of a whole genome in almost an afternoon these days because of it. And so this allows you to sequence tumors to look at molecular profiles. You can look at single cell sequencing, RNA sequencing of the brain to look at development, different cell populations as well too. And so with the advancement of this technology, it's opened up a lot of avenues for research. So whether we look at this omic analysis, so whether you look at genome, exome, which is looking at the DNA, transcriptome, RNA, epigenome, which is looking at histones, which is kind of like what our lab would be doing. This allows you to generate a lot of large data from biological research for analysis. However, when I came into the lab, and what we see a lot of these days is that bioinformatics is a bit of a black box. So a normal process that we see in our lab is that we take the samples, we send it to a sequencing facility, so they do all their work and they process the data and they spit out a bunch of images for us to kind of look at or data for us to analyze. And so we lose a lot of information in this process. And so what happens is that there's a bit of a disconnect between bioinformaticians and researchers, and the challenge is really the collaboration of these two professionals to carry out these next generation sequencing experiments. So to kind of look under that black box, so you would have your samples that you send for library prep and sequencing. These sequencing machines would spit out raw data for processing, which would be done by bioinformaticians who have the coding proficiency as well as the mathematical and the statistical background to do that. And they would be the ones spitting out the data that researchers with strong biological background could analyze and make sense of it. And sometimes there's a bit of feedback in terms of, well, they need to adjust this or show this or reanalyze it so that we can kind of answer the questions that we're trying to answer. And so a lot of challenge that we face in our lab, as well as other lab, is the collaboration between these professionals. And an example of this would be, so this was a paper that was published on our floor. And so as I was kind of reading through it, it is a bit of a complicated figure, and I was trying to make sense of it. And so when I look at the figure at the bottom, if you read it, it says TSNE visualization and 2D embedding showing constructed pseudotime trajectory of different lineages in the developing cerebellum. Cells within specific lineage clusters were selected, visualized using TSNE visualization, and then ordered based on reverse graph embedding method. Does that make sense to anyone? Not really, right? So because of this, it's hard for readers to really critically look at this literature and kind of see whether it really fits in with the hypothesis, whether the methods are rigorous enough that the data that they're getting is true to what it shows. And so because of this, what we've seen is that there is a bit of a reproducibility crisis in science. So there was an article published in Nature in 2016 showing that across science, there's about 70% of experiments that are not reproducible. There was also a study in 2012 showing that 6 out of 53 preclinical papers in cancer research is not reproducible as well. So that's terrible. And if you look at, so prime example would be this link that I show you over here. You guys can have, I urge you guys to kind of look at this. So this is a blog where one of the professors actually went through one of the papers that were published in Nature Biotechnology, which is a very high impact paper, and they show that the parameters and the analytical approach to the bioinformatics that they did was very flawed. However, the paper is still published there, and essentially no one can really say anything because no one really knows what's going on under the hood. And so the factors kind of contributing to this lack of reproducibility is selective reporting. So, you know, you look at your data and about 10% of it proves what you're trying to show, so you just show that 10% of the data. There's pressure to publish, which I felt as a grad student, and postdocs, even PIs feel it just to make sure that they can get grants to support their labs. There's bad analysis, not enough replicates, bad mentoring, bad methods, and the codes are unavailable. Fraud, as well as the technical expertise for reproduction also occurs, but that is a byproduct of bad method and code availability. And in my experience as a grad student, in this past 3 years, I've pretty much seen examples of all of those factors myself. And so our goal at BioBox was essentially to allow the researchers to have access to this raw data processing portion so that they can have, they can understand the method that was used to generate their data and be able to really understand how they got their results. And so, as I mentioned before, so it's to break the barrier to entry into bioinformatics. There's a lot of coding required right now, and so we want to design a software that researchers without any coding background in R or Python can kind of jump onto and start their analysis, which would allow reproducible workflow as we track all the parameters and the methods that they're using to do to their analysis, and provide an end-to-end platform for sequencing experiments. The other side of our focus is in education, so we wanted to foster literacy and critical thinking in bioinformatics. So one of the big components of our startup is integrating learning modules into our software. So, you know, we give tutorials on backgrounds in next generation sequencing, how to do analysis, how to make sense of it. And our goal is to really allow scientists to have access to this kind of research and be able to effectively use it for their projects. And if any of you guys take an HMB422, we're actually going to be piloting our software within this semester to allow the students to do some RNA sequencing analysis as well too. So I had a bit of a demo just to show what we can do. It's still in the early stages, obviously. I don't know if the audio will work.
Tutorial, we're going to be going over how to quickly get started, analyzing and visualizing your RNA-seq data, and then diving into some patho-enrichments that you can find. All right, so first we're going to go ahead and jump into our workspace. Find that in the side tab over here. And if you don't have a project, the first thing you can do is create this project. So I'm going to give it a name. Let's call it RNA-seq tutorial. And an organization that you want to use. So I'm just going to put in my personal org right now. And you're going to you're going to see this screen where you can start creating your bot boxes. Now, to start, just click this little new bot box button and boot up this new bot box. So the first thing you want to do is you want to load in some data. So you can actually load in data that's provided by the bot box team. This is an RNA-seq data set pulled from CHE, and we're going to load that into our workspace. And in this plot, I want to create a scatter plot. Okay, so it has some smart defaults. It's going to start plotting your log-to-fold change and your adjusted value. When you hover over each of these data points, you can get the annotation for what that data point represents. Now, there's a lot of data here, right? So sometimes you want to kind of zoom in a little bit. So you can pinch your touchpad or scroll in with your mouse, and you can zoom into the plot and click the plot itself to drag to reposition it. Okay. But you know, when we're looking at this is just everything plotted. It's not very useful. So let's go ahead and start filtering for some more insight. Okay, so we can filter by going into this little arrow button at the top of this data table. When you click it, you can specify your condition. So in this case, I'm going to say greater than 2 log2 change. Now it's up to you if you want to specify a second condition. You can do so here. But we're going to just run the plot for now. And when you're ready, hit OK. That's going to filter your data table, and it's also going to change the plot data points. Now notice what happens when we change this. It ran a pathway enrichment for you, and the results here, it says we have 13 insights, so these are the 13 pathways we picked up. And we can go ahead and kind of take a look at what we found from our genes that are greater than 2 log2 change. So here we have, endosomal molecular pathway, antibody-mediated complement activation, but it doesn't really tell us much. So what we can do to learn more about this pathway, we can actually expand it using this little arrow button. Now, there's not a lot of screen space, so if you want to kind of work with more room, you can, first you can hide this little sidebar, and these panels are actually resizable, so you can just hold and drag, and you can expand all of these panels. So let's go through this. So in this summary, we have some description of what this pathway represents, and a schematic. Now, it's a little small, and you can't really see the text. So what you can do is you can click the image itself, and it'll give you a full screen view. You can zoom in, you can kind of reposition it. But, okay, so let's get out of this for now. In this pathway, there's a lot of things happening, right? So it's hard to understand what the order of events is and what's going on under the hood. So what you can do is navigate to the Events tab, and we provide a step-by-step explanation, start to finish, of what's going on in the pathway. Now finally, you might want to see, okay, in my data set, of all the participants that act in this pathway, are there observations in my data set that match? So in fact, yes there is. So you can see them grouped here. That means in my data set, so in the plot points that I see in the bottom left, from this pathway, IGF2BP2 is found to be overexpressed, and BP3 is also found to be overexpressed. Now to learn more about what this gene is doing specifically in this pathway, again, you can expand that and you can read all about it here. Okay, so after we've done exploring all this, you can just go ahead and quickly save it, and it'll be saved under this project in this file box, and you can access this at any time. Okay, that about sums it up for today.
So it's a bit of a small portion of what we can do. So we're kind of developing more and more features as we kind of go. So you guys are very good coders, email us.
How much do you pay?
Competitive salary. So otherwise, kind of talking about my experiences working at a startup. So we're kind of working out of our condo right now. Obviously, it's very difficult to start, but what I've kind of gone through is there's a lot of endless pitching. So we have to go to investors and talk about what we're doing, why we're doing this, and why it's going to be fun, and why we're going to be successful. And so it's always a ton of presentation. I'm sure you've gone through yourself as well too. You have to be playing fluid roles, so be able to pivot. So you have to play multiple roles. So whether you're part of the development of the software itself, working on the business side, figuring out the accountings, dealing with legal battles. And so there's a bit of a flexible hours, but you have to put in a lot of time for these kind of startups to figure out. And one of the big challenges that we're actually facing right now is the idea of the intellectual property. So because all of us kind of started out as researchers at SickKids, now we're kind of in a limbo where SickKids is saying they actually own the software because we were part of that. We were part of them when we started, and so we're trying to figure out, how to get the license in terms of like equity and deals. So there's a lot of talk going on right now. It's kind of going well actually, but that's sort of the things that we weren't expecting when we were kind of starting this. It's not just coding and developing something cool and kind of succeed at it. There's a lot more nuances to it. But I think I've learned a lot from this experience, and if you have specific questions about it, I'll definitely kind of elaborate on it. So we've kind of joined in with UTest, which is an initiative at U of T for supporting early startups. So we're going to be doing a pitch for them this November as well too, and they'll be offering us kind of an office space for us to help us develop and inform this software. Just gonna end off with this. So I kind of wanted to end off with this slide because so going back, so kind of to give you context. So Christopher, which is my roommate and one of my fellow researchers, so he actually left his research position at SickKids to pursue this startup. And I've also recently made the decision to leave grad school as well to pursue a career in physiotherapy. And so we've kind of had this, hit this point in our life where we're kind of doing things that I guess other people don't expect. And I'm always getting questions on, why are you doing this? Like, why are you leaving this grad position? I'm sure Dr. G also had that question as well too, and my parents as well too. But there was an article published in Nature by someone that left grad school the first time. It kind of resonated with me, and I wanted to kind of read you guys something from it. So it says, earlier in my PhD, when experiments failed to support major hypotheses, I was shut down, unwilling to consider the possibility that embedded in the setbacks was an opportunity to try something new. These days, I still don't like getting negative data, and who does? But I have become more resilient, perceiving obstacles as essential to achieving growth and finding meaning. I know that many of my fellow scientists struggle to acknowledge missteps or vulnerability, as I once did, which makes it far too easy to feel alone in failing. This is why now I end my talks with "You are amazing" slide. So to be amazing does not require achieving recognition, awards, or major publications. To be amazing is to have the courage to embark on a scary, uncertain adventure and journey into the vast unknown. So as you guys are graduating, I hope you kind of think about different things. And I know there's the expectations to get into med school and to go into research. That's kind of what I felt going when I was graduating. But there is a lot of different options, and I hope you guys kind of take the time to think about what else you can do with your degree beyond those two major fields. And if you guys have any questions about my experiences or advices, you can definitely email me, and I'll send you guys the slide with my email on it as well too. But that's pretty much it.
Hey, do you have any general questions or?
Yeah.
I actually had a question about the BioBox software.
Yeah.
Because I do a lot of data analysis with my lab right now. But it's kind of like a separate question. Does it run offline? Because I'm curious how you guys pull the actual information from the genes.
It's all online. It's web-based. Yeah.
Okay, so
Yeah. You run it off your browser.
Are you scraping it from like Wikipedia or?
Sorry.
The information about the genes, where are you scraping it?
So we get it from Reactome. We're also accessing String Database. We also have access to OHM, and we just have a bunch of places that we got the license to get information from, and so we're kind of getting that and kind of integrating it in one place for researchers to look at. Yeah.
How long have you been going?
Sorry.
How long has BioBox been going?
So this is early this year, so around January. Even January, we were kind of figuring things out, talking about it. But I guess we officially formed a company early this summer. So now we've been kind of road mapping the software, putting it together, trying to find funding as well too. But yeah, we got some good interest from different labs and obviously the courses here as well. Yeah.
If you had to start over, what would be something that you would do differently?
Honestly, I wouldn't do anything different actually. when I think about like yeah, maybe I thought about, oh, maybe I shouldn't have started grad school, but honestly, when I think about it, all the experiences that I kind of gained through these past 3 years, they're so much more valuable than the degree that I'm not going to be getting by in this position. So I'm honestly very content with where I am, and I honestly don't think I would change anything.
Are there any specific skills that you feel like you either got to practice or that you learned in undergrad that help you now, or are there things that you wish you had in undergrad?
I think what I valued from undergrad was the stuff that I did outside classrooms. So,
Nice.
so I mean, you learn a lot in classroom as well, but kind of the lab, whether doing research, getting research experience, doing all the volunteer experiences, getting those kind of soft skills, that's what really I feel like kind of helped me succeed, or not succeed, at the next step of my career. So do other stuff outside. Yeah.
Did you have any startup experience, or you kind of just like got this idea?
We just jumped right into it. Honestly, we're still figuring it out as we go. Honestly, but we do have a lot of like mentors, and like the startup community is actually very open. So a lot of people are willing to kind of help out, give their inputs, and kind of talk about their experiences. So it was just a matter of kind of talking to a bunch of people that went through the same thing, and learning from their mistakes, also making our own mistakes, and kind of learning from it as well too. Yeah.
Do you have like someone with a business background working on a startup? Because you said no.
No, it was all like advice that we got from other people. But that's a significant proportion of the work actually. So it's a lot, not as much developing time, but more like trying to figure out the legal side of it, figuring out how to deal with equity in terms of like how much money we want to raise as well too, and all that kind of stuff we have to kind of figure out. And we're always thinking about it. Scaling is a big factor, how much everyone's getting paid. Like that stuff we kind of had to. There's a lot of good resources out there, obviously, and so it's a matter of kind of looking and doing for it, at least in our experience. I don't know if it's easy for you or not.
Reddit. I use Reddit.
Yeah. Yeah.
What's ideally the timeline for something like that? So a year from now, where would you?
So a year from now, obviously, we want to kind of have we're kind of facing a lot of glitches right now. So obviously, we want that kind of smoothed out. But once that's kind of okay, we want to start integrating it into actual research labs. So we do have some interest from the computational lab in SICKIDS, and so we're kind of getting feedback from them. And so once we kind of can put it together and really get our development streamlined, then hopefully by next year we can kind of get them to start using it in their actual research. Yeah.
In the slide, you did say something about financially stable.
Yeah.
So does that mean that all of the team is like savings or something?
So we had savings from our grad schools. It's really a regular part of it, honestly. At some point in the early stages, you do kind of have to inject your own money into it, and to really show that. And that's actually what a lot of the investors told us, that like even without that initial investment, like how far are you guys able to carry forward? And if you guys are able to show that you're able to succeed, it's gonna mean a lot early on. And in some ways, we're actually glad so we had some interest before, but we didn't take it because we didn't want to give away the equity. And so by doing that, we kind of got to keep more of our company and kind of run it the way we wanted to build it as well too. And again, that's something to kind of consider going into the future for us as well. Question back there, yeah.
When you entered grad school, did you feel pressure to go the research route? Is that why you entered originally? And during grad school, what made you want to start this startup originally and leave the researcher field?
So in terms of going into research, I think I did that. I did like research. I still love research, honestly. But I think it was like, it felt just like the logical next step to do it because like everyone seemed to be doing it, and maybe not the best mentality in the end, but that's pretty much the reason why I kind of went into it. And it just made sense for me. I felt like there was nothing else that I could really consider. I didn't get into med school after undergrad either, so that's why I did it. What kind of, I guess, compelled me to do this startup was because of the way the field was. So there's a lot of bad research going on right now. Not that I'm saying I'm amazing either, but there's a lot of in terms of the bioinformatics side, we've tried to replicate a lot of different studies, and it's just it doesn't happen, and we've seen a lot of fraud and deliberate hiding of research, whether it's because they kind of had to do it that way, or they were pressured to publish, or that was just what they needed to get to the big impact. So we wanted to kind of take initiative and provide a solution that can kind of combat that.
How much do you think having a team helped you instead of, for example, going at this alone or contracting someone to do the software?
Oh, team was very important, and it kind of fell into the right place because everyone kind of had different experiences that came together. And honestly, team chemistry, we learned from a lot of people that did the startup or had startup experience is very important because of kind of the unpredictability of it all. So whether it's the lowest of the lows or the highest of the highs, it's kind of being able to stay committed and kind of trusting each other. That's why it's really, I guess, important having a team when we're starting.
Speaking about research and about papers and bad papers, how would you just look at a paper and go, wow, this is a terrible paper, and then versus a paper you go, you know what, I know this is done right. How would you be able to tell some signs?
On the surface, it's hard. You actually have to really dig into it, and again, it's because it's part of the reason of reporting it, right? So when you look at methods, for example, for these kind of bioinformatic papers, they would just say, oh, we just use this software. They don't really talk about the code they use, the parameters they set, and that really changes the way the output, what the output of the experiment is. And I know a lot of labs adjust their parameters until they get the output they want, and they publish that. So that's kind of what's happening. The dirty little secret in the field. Yeah.
Okay, so if I can recommend, let's take a quick 7-minute health break, come back, we'll get the Z set up, and then we'll continue on with the discussion of what you might want to do next. All right, thank you.